Good morning and hello everybody around the world. And thank you for joining another presentation of our second webinar series, Hoppy Topics. Today we talk about our breeding program and recently launched Varieties. My name is Frank Pfeiffer and I'm the technical director at Hopsteiner here in Germany. For everybody who's joining us for the first time, I would like to give a very short introduction. Our webinar is a free and live online seminar with presentations about various hop-related topics. All of us have limited possibilities to travel and meet each other, and therefore we would like to additionally offer this kind of online support. In this way, we want to keep you up to date about latest research in hops. Before we start with the presentation, I would like to point out that we will be happy to receive your questions and comments. Your microphone and camera remain switched off during the webinar, but you can address your questions at any time through the Q&A tab on the control panel. I will pick up your questions at the end of the presentation. The presentation will take about 35 minutes, followed by the mentioned Q&A session, so in total we have about 1 hour time. So coming now to our today's presenter. So today's presentation will be held by Dr. Alexander Feiner, our expert in hop breeding and, to be honest, in skiing as well. Alex already grew up on a hop farm in the Hallertau, and so he studied agricultural management at the University of Applied Science Weinstefan in Germany. After joining a 6-month internship at the Hopsteiner research station in Yakima, USA, he finished his Master of Science in 2012. In the same year, Alex joined our Hopsteiner team and became the head of our breeding in Europe. In 2014, he started his PhD as an external student at Martin Luther University Halle and Leibniz Institute for Plant Biochemistry, working on metabolomics and genetics in hops, and finished with his doctorate degree in January 2020. Alex, we are curious about more insights of your work now, and we especially the outcome, namely Akoya and Solero. So it's your turn now. Have fun.
Thank you very much, Frank, for your kind introduction. And welcome everybody to our today's webinar. And as Frank mentioned, I will in the next 35 minutes, I will focus on Solero, Aqua, and what we have else in the breeding program in the pipeline. And the following questions I would like to answer in our webinar is why we do breeding, how we do breeding, and what is actually in the pipeline, what has been released, and what do we do. First of all, I would like to introduce the breeding team, which is active in our program. We have 2 main locations. The main location is Yakima, USA, where we have greenhouses, nursery, labs. And the main difference to Europe is that we are also a grower on 4 farms in the Yakima Valley. And since 2012, we have a new breeding location in Mainburg, Germany, where I'm located, where we also have the same setup with greenhouses, nursery and labs. But the main difference, as mentioned, is that we work with contract growers, and we are not our own grower here in Europe. The breeding team consists of Nikolai Pitra, the head of breeding in Yakima. I'm responsible for Europe, and since beginning of the year, we have also Martin Wallinger with our team and Dr. Paul Matthews as our senior researcher. The Hopsteiner breeding program started in the late 90s in the US because there was a massive powdery mildew problem and there were no resistant varieties present at that time. And therefore our Hopsteiner breeding program started because we wanted to breed new high alpha varieties which are resistant to powdery mildew. And the first varieties were released in 2004, 2006, Apollo, Bravo, and Super Galena. At that time, the US was the high alpha producer, and therefore these varieties were our high performing varieties. With 2013, we expanded the breeding program with a location here in Mainburg, where we apply actually the knowledge which has been gathered in the US in the past decade, and to breed varieties for the European market. In the years 2010 to 2020, we released varieties like Delta, Calypso, Lemon Drop, Eureka, Lotus, Sultana, mainly for the craft beer industry, but also for the big ones. And just recently, Altus and Contessa. Altus is a high alpha dual purpose hop, and Contessa a hop with noble aromas. And with 2020, we have now the first varieties in the field, which are named Akoya and Solero, and I will talk about them in the next few minutes. Here's an overview of our released varieties. As mentioned, Super Galena, Bravo, Apollo as high alphas, and Eureka, Lemon Drop, Calypso, Sultana, Lotus, with very unique aroma profiles for really good beers with hop forward aromas, and just recently released Aqua in Germany and Solero. This slide shows the goals in a breeding program. The goals here are quite generic. They are present in every breeding program, or they need to be applied in every breeding program for each crop, because at the end what we want to have is yield and quality. But yield and quality is only possible with certain resistances to abiotic and biotic stress. So these three goals, they interact together. There's no quality without resistance, and there is no yield without quality. And this drives our daily business to breed varieties with higher resistances. And this is a fact which is present since many years, and the projection is that we have to live with that fact. It's called climate change, and everybody knows it. Here on this slide, you see the temperatures, the average temperatures in Germany in the last centuries. And you see from the 1880s to 1970s, it was kind of normal that the average temperatures, they fluctuate around a certain average value. But since the 70s, late 80s, we see a clear trend and a clear increase in average temperature. And all the colored lines here, they are different climate models projecting temperature in the future. And every single model shows us that in the next century, we have to live with higher temperatures and to live with also less precipitation, but also with a global warming. And this drives our daily business, because brewers and growers, they need varieties with a higher reliability, which can better handle the climate change and deliver stable yields and stable alphas. And this is another fact. It's called the European Green Deal. Two strategies were released. It's a biodiversity strategy, which addresses the conversion of at least 30% of the European land and seas into effective protected areas, which means there won't be as much as acreage available as today. And another strategy is the farm to fork strategy, which says that chemical pesticides needs to be reduced by 50% by 2030, and the reduction of nutrient losses needs to be reduced while ensuring the soil fertility. But also then this means that fertilizer applications needs to be reduced by 2030. And these are facts which drives our business, and we summarized all the requirements for new varieties in that table. We have two categories of new varieties. One category is the replacements, where we talk about aroma varieties, which we want to substitute, like Perle, traditional, and high alpha varieties like Hercules. But they all need to have, or all need to meet certain requirements. They need to have a certain yield. For aromas, we need to have at least 2,500 kilograms per hectare. For high alphas, more than 500 kilograms of alpha per hectare. And they all need to have resistance or tolerance to abiotic and biotic stress. And what we also need for them is pleasant bitterness quality. And when we talk about replacement for aromas, the aroma needs to be traditional and comparable. The second category is unique flavor, like citrusy, fruity. All these varieties which are in the unique category, they also need to meet our requirements of yield and resistance, but the aroma needs to be unique. And our focus is that first, the agronomic characteristics needs to be there and needs to fit in our requirements. And second are the brew trials. So first agronomics, second brew trials. And for those, I think most of you are quite familiar with hops and how they look like. But here just a short overview about the species, Humulus lupulus, which belongs to the family of the Cannabiceae and the order of the Rosales. Besides Humulus lupulus, there are two more species out there. It's Humulus scandens, an annual version, and Humulus yunanensis, and only Humulus lupulus is of commercial interest. It's a diploid species with quite a large genome of 2.8 gigabase pairs, very complex, large genome, very tough to study. And it's a perennial crop, and as you see here on the pictures, it's dioecious, and only the female cones are cultivated, but we also need the males for breeding purpose. About 95% of the global hop production is consumed by the beer industry. And in 2020, we have nearly 63,000 hectares on a global scale. And we estimate the crop around 130,000 metric tons, which is quite a good crop, which is out there. Hop harvest has started last week. It looks quite good. And yeah, we will see if we can meet the estimation. How long does it take to breed a variety? Every year, in year 0, we start with a selection of parents. So we choose the best females, the best males. We cross them, and then we get the seeds. We germinate the seeds. We screen the seedlings. And each year we start with about 50 crosses, 25,000 seedlings. In year 2 to 3, we screen them on the so-called single hill evaluation, where we check for disease resistance, for chemical traits and maturity. And from the 25,000 plants, only 7,000 make it in that step. And out of that step into multi-hill evaluation, only 20 to 30 plants make it. And multi-hill means we check the agronomic traits, the chemical traits on different environments. And we do also the first brewing trials. And out of these 20, 30 plants, only a few make it to the semi-commercial step, where we check, or where we want to confirm the agronomic and chemical traits, and where we do extensive brewing tests. And if you are lucky, after 10 years, you might have a variety or not. So it's there's a certain proportion of luck in there. But this breeding cycle starts every year again. So we have overlapping and multiple breeding cycles. So you cannot say every 10 years we can release a variety, because we have overlapping breeding cycles. and to speed up in that process, because 10 years is quite a long time, and we have new requirements, new pests and diseases out there. So we need to be quicker in releasing new, efficient and high-performing varieties. And therefore, what we do at Hopsteiner, we apply molecular breeding and new technologies, where we do massive phenotyping and genotyping and massive computing to breed new varieties in a more efficient way, in a more targeted way. And this molecular selection tool is applied in our program, and this helps us to speed up the breeding cycle about 2 to 3 years. And this is quite a good improvement for us. And what does this actually mean? And a short explanation about the genome-wide association study. What we do there, first of all, you make a reference genome. You extract DNA out of hops, and then you create a reference genome. And up to now, we have quite a good one, which we can apply. And then you have plants cases with disease. So unhappy plants, diseased, they have powdery mildew or downy mildew, and we phenotype them, we screen them, and then we extract the DNA of them and we sequence them. And we have cases without disease, quite happy, they look good, they are green, no disease on them, and we also extract DNA of them. And what we do with genome-wide association is we want to find the association between the genetic variation and the observable trait. This tells us which part of the genome is associated, for example, with a certain resistance to powdery mildew or downy mildew. And these genetic markers can be then applied as a selection tool and helps us to quicker choose new parents and also offsprings. And where are we now? What is the status quo in Germany in 2020? In the so-called multi-hill, where we have 7 plants per experimental, we have currently 74 experimentals. And they are all assessed and screened for their agronomic and chemical traits. And we do also the first brewing trials, and we evaluate the aroma and the bittering quality of them. And in row plots, which means 100 plants per experimental, we have 7 on multiple locations, and we do multiple brewing trials with them, and also the first yield calculation. And with the year 2020, we have now Akoya and Solero in the fields. We already applied for an EU variety registration, and we have 2 varieties out there, and I will talk about them in the next few slides. To better assess varieties, we created this Hopsteiner flavor wheel with 8 categories, from citrusy, fruity, floral, herbal, spicy, resinous, sugar-like, miscellaneous. And all these categories and descriptors, they help us to categorize a variety and to better define the aroma profile. And here's an example of an experimental which was advanced to row plots, to the 100 plants per experimental. It's an aroma variety, aroma experimental, and the special feature is it could be an alternative for fine and noble aroma. So in the category of Hallertau, Mittelfrüh, Hersbruck, in that category, it has a alpha acid content 4 to 6 and kind of a spicy, mild aroma. Quite resistant, good yielding, and I'm really curious about the future of that one. We already performed some brewing trials and they were really promising, and I hope to do more with that experimental. Another one, 236, 234. It's also an aroma, and the special feature is new aroma, unique aroma. It's cascades offspring and has 7 to 9% alpha, citrusy, mint, fruity, also berry notes, very unique and also very resistant, good yielding. The first brewing trials were quite promising and really unique aroma profile in dry hopping. And yeah, I'm very excited to do more with that in beer. And that's also a special one, the last one we have in row plot, which is really promising. It's a high alpha, but also a high alpha with unique aroma. It's green apple. It's clearly green, fresh apple. We picked the first variety, the first wine yesterday, and we got already 16-17% alpha. So it's quite high. It's not even ripe right now, so we see higher alphas coming in, and it will be harvested, I think, next week. But already now there is the green apple aroma, sugar-like. It's just an unique, outstanding variety with high contents of the known esters, ketones, and terpenes. It's a bomb. It's an aroma bomb, and very curious to work with that in the future. And now I would like to introduce you our new varieties, our new releases. First one is Solero, released just recently, and the profile is really in the citrusy, fruity, sugar-like area category. It's also an offspring of Cascade, and the variety can be described as unique aroma variety with the aromas of tropical fruits, mango, passion fruit, very outstanding aroma, and yield is quite good, between 2,500 2,800 kilograms per hectare and more. It's late ripening. It will be one of the last varieties to pick, but the good resistances to diseases still keeps it green out there. No hits on them. Looks quite good, and the alpha content is between 8 and 10, something typical for an aroma variety. And now I have some videos for you. I hope it works with your streaming quality. This is a baby hop field from Solero, planted this year in spring from rhizomes, and we already expect a crop in that baby hop field. You see the huge amount of cones on there. It looks really good. All wines made it to the top of the wire. Got nearly no sprays with chemical protection. It's a kind of an outstanding plot here. I'm very happy about the performance here. And here's a video of first year Solero. What is really typical for that variety, you see brownish leaves down here and yellow leaves. The leaves turn yellow and about yellow early July. This is genetically driven. The variety kicks off all the leaves down low, which are not necessary to produce more yield. It only concentrates on the top of the vine to guarantee a good yield. And as you see here, the top of the vines, they are loaded with cones. It has quite large and long side arms, but this does not really affect yield, because they just hang down there and they don't grow together. So the canopy is quite good, and it doesn't shade out and guarantees the yield here on top. And therefore I'm quite happy about the performance of that first year Solero. It shows very typical profiles and characteristic in this year compared to last year's, very stable. And that's the second one. I'm now talking about Aqua. Aqua has I need my laser pointer again. Aqua has a slightly citrusy, floral, herbal, spicy characteristic with aroma, with alpha acids between 9, 10, 11, and oils between 1.5 to 2. So quite quite a little bit higher than other varieties, but the aroma is tea, hoppy green fruits. So a very classic aroma variety with herbal and tea-like, and as well slightly fruity with mint and pepper notes. Very similar to existing varieties, Perle or Hallertau traditional. Brewers in the last few years, when they came to Mainburg for evaluation, we had the varieties on the table and we compared them to existing aroma varieties, and they were quite happy about the profile. It's very comparable to Perle and tradition. And the yields is about 2,300 to 2,400 kilograms per hectare and more. I just got the yield data from this year. It's a little bit higher than here. We need to see if this is stable on each plot, but we are quite happy with the yield performance and also with the alpha performance. The morphology of the plant is cylindrical, slightly club-shaped, medium to long side arms, a little bit like Solero, but not that club-shaped. And the cones are compact, but slightly elongated. And the interesting part about Akoya is the flowering and the maturity time. It's early to medium flowering from time. The flowering time is early to medium, but what is interesting is the very short maturation. So from flowering to the ripe cone, it's very fast. It really switches the time, or it switches from flowering to being ripe, and maturity time is early. And as I mentioned, Aqua is already harvested on some plots. It's early ripening, and this is what we need, because if we want to replace existing varieties which are under pressure because of climate change, we need to have something which is kind of early ripening. And also for Aqua, I have some videos for you to get some impression how Aqua looks in the field. And as mentioned, Aqua is a little bit club-shaped, but this does not really affect the yield, because as you can see here, there is enough light in the field. When we fly over there with a drone, you see it's not shaded out. The cones can be established and get to full ripening. It's quite, and the whole plot is very uniform. And also here, this is a baby hop field. These videos were made 2 or 3 weeks ago, and the next video you will see what happened in the last 2 weeks. This is the baby hop field, and here is the harvest of Aqua yesterday in that field. You see here the vines are loaded. I'll play it again. The vines are loaded with cones, very big ones here. And it was really fun to harvest Aqua yesterday. Also, alpha contents look quite good. Yeah, we will see what will be the end of that story this year, but yield looks good and alpha content looks also quite stable. And this is a video of the baby hops I took yesterday. The video. I'm quite curious about the yield in that plot. Here again. And comparing these to the pictures I showed before, in the last 2 weeks, there happened a lot. Yeah, quite curious about that yield. And here a table about the number of plant protection treatments, and that's what I mentioned before. The new releases are quite resistant and tolerant. We compared it to the Aroma variety Perle. For downy mildew, we see that Perle was an average sprayed about 3 to 5 times for downy mildew, Akoya 2 to 3 times, but most of the sprays were like prophylactic. There was no need to spray. The farmer was like, yeah, it was during blooming time. He wanted to secure the crop without any pressure, and but definitely with 2 to 3 sprays, you're fine. Solero got 1 to 2, so very stable varieties. And for powdery mildew, Aqua, we had some fields they got no spray, and also Solero they got no spray for powdery mildew, and they are still safe, and there was no powdery in that Aqua field, which made me quite happy. For aphids and spider mites, yeah, they are kind of comparable, depending on the location. On more sandy soil, there is higher pressure, but in general, there is nothing which is outstanding, I would say, and very comparable. So that's about the varieties. And now the next few slides, I would like to talk about the evaluation of the breeding lines in beer. It's tough to judge the variety just in the fields where you smell the green and fresh aroma. The next step is then the dry hop. This is another, yeah, it's closer to the aroma which can be delivered to beer, but at the end you need to evaluate the aroma and the profile in beer. And for that reason, we got a new experimental brewery in Mainburg to answer questions in our breeding program, but also other questions of new products, whatever. And we got a 5 hectoliter Braucon brewery, steam heated, internal boiler. We have mash and watt kettle, lauter tun and whirlpool. So we can answer every question here now in Mainburg in terms of evaluating new breeding lines, new varieties, new products. And for Aqua, we made beer, a Bavarian lager, was single hopped with Perle. The one beer was single hopped with Perle, and the other one exclusively with Aqua. And we had a big tasting at the Braubeviale in Nuremberg, where more than 300 tasters evaluated both beers. And we wanted to answer the question, are the varieties similar? Are they comparable? And therefore, we asked the taster, can you evaluate the bitterness quality and the bitterness intensity and the overall expression? And what we saw is, in terms of bitter quality, there was no difference. There was a slight difference in bitter intensity because also the IBUs in that Aqua beer were slightly higher. Told us that because we gave the same alpha rate, that maybe the efficiency of Aqua is slightly higher, and we got slightly higher IBUs, and it was therefore slightly higher assessed as with a higher bitterness intensity. But the overall impression was the same of Aqua and of Perle, and this made us very happy, because when the impression is the same, it's comparable. And also we asked for the preference. Do you like more the Perle beer or the Aqua beer? There was also no significant difference, a slight tendency to Aqua, but yeah, no significance. And these results made us very happy. And based on all the agronomic traits and on the brew trials, we did also massive brew trials with other breweries. This led then to the decision, we can release that variety, we are safe, this is a very stable variety, delivers stable yields, alphas, and also performs in beer, and therefore the story of Aqua was done, or was perfect, and released it. Therefore, now I'm at the end of my presentation, and I would like to summarize what I was talking about today. I wanted to answer the question, why do we breed? And we breed because of climate change with rising temperatures. We have reduced use of plant protection and fertilizer in the future. We are required to reduce that. And for that, we have breeding locations in the US and in Germany, where we want to answer all these questions, and where we are actually doing the research. And we see quite a big acceptance, high acceptance of our new varieties in the brewing industry. And this makes us to expand the acreage in the next years, because we see there's a need for these varieties in the future on the brewer side, but also on the grower side. And Akoya and Solero will be followed by new other varieties in the future. So it's just the beginning here in Germany. We already released a bunch of varieties in the US. Our colleagues in the US do the same job as here, and there will be also new varieties in the US answering that questions. And yeah, at the end, our Hopsteiner research program is an essential part of securing a sustainable hop production in the future. We have many projects in the pipeline. We want to do more in the future. We will release more varieties and hopefully do more applied research in the future. My last words are here. I would like to thank the hop growers being involved in the Hopsteiner breeding program. There are 5 growers involved, and they do a really good job. Otherwise, we couldn't have released a variety within that period of time of 8 years. So they do a really great job. It makes really fun to work with them, and thank you so much for your support. And now I'm ready for the Q&A session. I thank you for listening today, and I'm more than happy to answer your questions.
So Alex, from my side, thank you very much for this fantastic presentation. For me, it was more than a presentation, it was a demonstration of your work, and we all had the feeling that you are the father of these new varieties. These are your babies, and more babies will coming up soon. So I will read now the questions coming in, and you will then be able to answer accordingly. So we got one question here that comes in from James, and he's asking, do you work on the next generation of high alpha varieties?
Yes, exactly. Good question. Of course, as I mentioned at the beginning of my presentation, we try to find replacements and substitutes for existing varieties. We see a need also in the high alpha sector with more and better resistances to powdery and downy mildew. It's tough to breed for new high alphas because the production of alpha is genetically linked or closely linked to a susceptibility of powdery mildew and downy mildew. But we are working on that. We are quite promising breeding lines in the pipeline, and maybe in a few years we have also the answer for a new high alpha.
Good. Next questions come in now from Michael, and he's asking for, do we have any collaboration with universities or with institutes?
Yes, in the US and here in Germany, we collaborate with universities and institutes to answer certain questions. Our powdery mildew studies or my downy mildew study was in collaboration, as you mentioned, with the Institute of Plant Biochemistry in Halle with the IPB, and we do many collaborations because we want to collaborate with the experts in a certain field, and therefore we need to collaborate because we don't have also the possibilities to to invest in all the machines. They are quite expensive, and it's always better to work with the experts in such a field.
Absolutely. So more requests, some more questions coming in. There's one from Andrea, and she's asking about the verticillium build. Is there any resistance of Akoya and Solero, especially in the lesion form?
It's a very good question, Andrea. We see that the verticillium build is getting a problem more and more every year, and this year is a verticillium year. In the Hallertau, we see many fields breaking down because of that fungus, and we tested Aqua for the lethal strain for non-alfalfa, and it's moderate. The resistance is moderate, so there is a tolerance to that, and this can also be seen in the field this year. We planted Aqua on many fields where there was a problem with verticillium before, and we see exactly what we got in our tests. It's moderate. We see it can be grown in such a field, but if the pressure gets too high, if there was in the years before a heavy infection, also plants of Aqua break down, but it's just a few of them, and therefore we see that maybe a crop rotation in that field of a break of 1 to 2 years, and then planting Aqua might then not guarantee, but this might then lead to a cultivation again without verticillium build. With Solero, we see kind of the same effects in the field, but the test needs to be done with Solero, and we haven't expanded Solero on that extent like Aqua, and therefore we need to gather more results in the future. But we also see that there's a quite resistance or tolerance to verticillium also in Solero, but we are not that advanced with the results right now.
So we are looking on our screen. There are some more questions coming in, and
so far seems like no more questions. Huh?
Yes, all people are happy now, and we have seen that there are still some problems outside to solve, especially with new varieties, and that is also yes, our intention to keep an eye on this in the future and to be able to.
Now here's one more.
To bring more and more nice and good hop varieties as a replacement for the existing ones. So there's a question from Silvana, and she's asking, hi Alex, you mentioned the need of plants which are more tolerant to drought or reduced water ability. How is this implemented in your breeding program, or have you or could you also check with genomic approaches?
The last few years, especially in the Hallertau, they were really good years to screen for, I would not say drought resistance, but definitely for a tolerance against high temperatures and less precipitation. So we do not screen in greenhouses where we like treat plants with less water, but what we can do is we have some genetic markers for drought resistance. We established them in the US. Our colleagues did that in a collaboration, and we could apply these genetic markers, but also then the screening in the field tells us if they are more stable. And as I mentioned, the last few years they were really good years to select plants like that. And Solero and Aqua gave us the stable yields and comparable yields to other varieties or even higher, and therefore we mainly screen in the field, and we don't do screening in the greenhouse.
Thank you, Alex. Next question is coming in, it's from Dirk. So Dirk is asking if we can recommend Solero for dry hopping, and if yes, what is the recommendation regarding the amounts?
Yeah, definitely Solero is a variety which is recommended for dry hopping with tropical fruits, like passion fruit, mango, and citrusy notes. What we have seen is that variety has an astronomic high amount of isobutyrate, which is also present in other fruits, which gives the fruity character, and it's like 5-6 times higher than in other varieties. And this gives you the very distinct and unique aroma profile. And we could prove this in different beers, and recommended amounts. You get some notes already at 200-300 grams per hectoliter, depending on what you want. If you really want, but if you want to have the punch and this really fruity taste in your IPA pale ale, I would recommend at least 500. I would, in our tests, in the next test, what we want to do, or in our screening, we already we extended the amount to 700 grams per hectoliter. So you easily apply 700 grams per hectoliter. You cannot overdo it. It's just then what you really want in your beer, but something in there. Last year at Brau we had this this IPA, yeah, this double. Yeah. No, it was double IPA. IPA was it? It was double IPA. It was a double IPA, yeah. And I think in that beer we added more than 2 kilograms per hectoliter. So you cannot overdo it. Use as much as possible.
Yes, Alex, thank you very much for your answers. And now let's have a look there on the board. We have currently no more questions coming up, and so we think that you are happy now with the given answers and also with the presentation as well. And okay, now let's close the session for today. And on our website hopsteiner.com you can find some related papers about today's topic, and also you can check out the section technical publications. So there are a lot of different other topics there as well from the last years of our research and development programs and the released papers as well. After closing the webinar, you have the chance to give us a short feedback by answering 2 short questions, if you like to do so. In addition, you will receive an email including a link to watch the recorded video of today's session. And finally, I would like to say that we have the next webinar on the 1st of October. That is a presentation from myself with the title Hop Flavor and the Stability During Shelf Life of a Beer. Thank you very much for joining our webinar, and we hope you enjoyed the time with us today. We are looking forward to meeting you soon again, and all please stay healthy and goodbye.
Thanks. Bye-bye.